हेलो स्टूडेंट्स, चैप्टर नंबर 3 ह्यूमन रिप्रोडक्शन में हम लोग पढ़ रहे थे गैमेटोजेनेसिस। फॉर्मेशन ऑफ गैमेट्स को कहा गया था गैमेटोजेनेसिस। इसमें लास्ट क्लास में हम लोगों ने पढ़ा था स्पर्मेटोजेनेसिस। मेल इंडिविजुअल के अंदर, मेल ह्यूमन बॉडी के अंदर स्पर्म का फॉर्मेशन कैसे होता है, इसे हमने समझा था लास्ट क्लास में। और स्पर्म के बनने की प्रोसेस को हमने कहा था स्पर्मेटोजेनेसिस। आज हम फीमेल इंडिविजुअल में ओवा का फॉर्मेशन कैसे होता है इसे समझेंगे। और ओवा के फॉर्मेशन की जो प्रोसेस होती है या ओवम के फॉर्मेशन की जो प्रोसेस होती है इसे कहा जाता है oogenesis। तो अब हम समझते हैं oogenesis की प्रोसेस को।
Primary oocyte consists of 44 XX chromosomes. It is around 35 microns in diameter. As the follicle undergoes maturation, primary oocyte increases in size and reaches about 140 microns in diameter. The flat follicular cells now becomes cuboidal. Follicular cells multiply to form several layers of cells around the developing primary oocyte. Scattered amorphous material accumulates between the inner layer of follicular cells and the primary oocyte. This glycoprotein-rich material fuses to form a complete striated membrane called as zona pellucida. It is derived from both primary oocyte and follicular cells. Numerous microvilli of the primary oocyte projects into the zona pellucida, where it comes in contact with similar processes of inner layer of follicular cells. It helps in transfer of nutrition to the developing ovum. The multiplication of follicular cells gains pace, and as a result, small spaces appear in between the follicular cells. These spaces fuse with each other, forming antrum folliculi. It is filled with a fluid called as liquor folliculi. The follicular cells which forms the outer layer is called as membrana granulosa. The follicular cells that intervene between the antrum folliculi and zona pellucida is known as cumulus euphoricus or cumulus ovaricus. Those follicular cells that suspend the zona pellucida and developing ovum constitutes the discus proligerus. At the same time, changes take place in the stromal cells of ovary surrounding the developing follicle. These stromal cells organizes to form a sheath known as theca folliculi. Theca folliculi consists of two layers. Theca interna, inner vascular and cellular layer. Theca externa, outer fibrous layer. At this stage, the primary oocyte completes the first meiotic division to form secondary oocyte and first polar body.
The process of development of a mature female gamete, that is, the ovum or the egg cell, is called the oogenesis. And this process, remember, it is going to begin in the embryonic stage. Unlike in the male, the process begins in puberty. This process, oogenesis, is going to begin in the embryonic stage in case of the female. Now, the ovary is going to contain around 2 to 4 million of the eggs, immature egg, at the fetal stage itself, right? And nothing is going to be added. No more eggs are going to be added up after the birth. So, whatever we have at the fetal stage, that continues at the later point of development. The egg in the fetus is called the oogonia, and that is the gamete mother cell. And all the development that we're going to talk about is going to begin with the process of oogonia. Let us now understand the process of the oogenesis in the females. Now, the oogonia undergoes meiotic cell division, and during the prophase 1 of the meiosis 1, the developmental process is going to be temporarily arrested. That is, there is no more development after this particular prophase 1 stage. And at this stage, the oogonia is called the primary oocyte. So, the oogonia arrested at the prophase 1 is called primary oocyte. Now, the primary oocyte gets surrounded by certain granulosa cells, and now it is called the primary follicle. So at the time of puberty, the number of primary follicle is going to be around 60 to 80,000 per ovary. Now this primary follicle is going to undergo further development during the puberty, and they get additional layers of granulosa cells surrounding the primary follicle, and now they also form a new layer, new theca, and that is called the secondary follicle. Now the secondary follicle develops into tertiary follicle as a fluid filled cavity called antrum is developed in the secondary follicle. Now remember, this is also referred to as antral follicle, and the theca that we are talking about at the secondary follicle stage, now they get organized into theca interna, that is the internal theca, and theca externa, that is the external theca. So to summarize, you have the primary oocyte surrounded by granulosa cells called a primary follicle. The primary follicle develops into secondary follicle as and when there is additional layer of granulosa cells, and the secondary follicle develops into tertiary follicle when there is a fluid filled space called the antrum, and also known as the antral follicle. Now the primary oocyte within the tertiary follicle. So till now what we have understood, that the follicle is developing, and inside that follicle we are having the primary oocyte. So now the primary oocyte within the tertiary follicle during the puberty, it completes the meiosis 1. So as we understood in the previous slide, that it is in the prophase 1 of meiosis 1 that the development is arrested. So now what happens is the development continues of the primary oocyte, and the development continues by completion of the meiosis 1. And now the meiosis 1 is not an equal division, it is an unequal division, and it is going to occur when there is an elevated level of the luteinizing hormone, or the LH, also known as the LH surge. So in response to this LH surge, there is going to be the completion of meiosis 1 of the primary oocyte. But remember one thing very important here, the division is unequal, that is you're going to get a larger cell and a smaller cell. So the larger cell is haploid, both of these are haploid, so the larger haploid cell is called a secondary oocyte, whereas the smaller cell, the tiny cell, which is also haploid in nature, is called the polar body, first polar body, or the polar body 1. Now the tertiary follicle that has developed, right, till now we talked about the tertiary follicle. Remember, two things are going on simultaneously. One is the follicular development, and inside that the oocyte development. So the primary oocyte completed its meiosis 1 and gave the secondary oocyte. Now outside the secondary oocyte is the tertiary follicle. Now this tertiary follicle at this time matures into Graphene follicle, and the Graphene follicle is going to get another layer, a new membrane is formed, the zona pellucida. So this new layer, new membrane around the Graphene follicle is called zona pellucida. Now remember, at this stage when you are going to have the Graphene follicle and you are going to have the zona pellucida with the secondary oocyte, at this stage the ovulation occurs. Remember, the ovulation is going to occur again in response to a change in the hormonal level, and that is nothing but the surge in the luteinizing hormone level. And during the ovulation process, there is going to be the rupturing of the Graphene follicle or the mature follicle, and the ovum is released surrounded by a layer called corona radiata. We'll see that in a minute. Now the meiosis 2 is completed when a sperm penetrates its plasma membrane. Remember, till now what has happened? The secondary oocyte has completed only the meiosis 1. There is no meiosis 2 as of now. So that is going to be completed only when a sperm is going to penetrate its plasma membrane. And only after the completion of meiosis 2 is the egg technically called an ovum. So we are using the ovum loosely, but the word actually should be used only after the completion of the meiosis 2. Now let us have a look at the Graphene follicle under a microscope. This is the fluid filled space, and this must be the antrum, and here we are going to have the secondary oocyte that has completed the meiosis 1. Now let us understand what we spoke with the help of a diagram. So as you can see here, this is the cortex region of the ovary, and you can see these are the primary follicles. These are the primary follicles, and these primary follicles develops into the secondary follicles, tertiary follicle, right, and then the mature Graphene follicle. And during the process there is also development of the oocyte. So as we discussed, there is going to be the addition of granulosa cells just outside the oocyte, primary oocyte, and over time when it is in the tertiary follicle stage, there is going to be the formation of this fluid filled space, and this is called the antrum, and that's the name antral follicle or the tertiary follicle. And from here the next stage, that is going to be the mature follicle or the Graphene follicle, and you can clearly see the well developed structure, the space, fluid filled space called the antrum, along with the oocyte, and this is going to be the secondary oocyte stage. And also you can see the layer called zona pellucida. And from here during ovulation, what happens is there is going to be a rupture of the mature Graphene follicle and release of the ovulated oocyte or the secondary oocyte, along with a layer, as I said, there is the corona radiata. Now we will see what these structures are, that is corpus luteum, and under what condition does it actually develop. That we'll talk in the upcoming concept, that is menstruation. Now let us summarize the entire process of oogenesis. So the oogenesis is going to begin with the oogonium that is undergoing the mitosis to increase the number. There is going to be the primary oocyte and the meiosis 1 begin. Now this primary oocyte is going to get arrested at the prophase 1 of meiosis 1, and all of this event is going to take place before the birth. And after birth, the process of development continues only after the puberty, and that is going to resume the meiosis 1. So the oocyte meiosis arrest at the metaphase 2. That is, it is going to develop, that primary oocyte is going to develop into secondary oocyte and the first polar body. Remember, this is a meiosis 1 and this is an unequal division. Now after the sperm penetration, there is going to be the completion of the meiosis 2, and you are going to get another unequal division with a larger mature ovum and the second polar body. Right, now the first polar body also divides and produces 2 more polar bodies, and all of these polar bodies are going to get degenerated. So you can see a larger cell, that is a mature ovum, and a smaller cell called the polar bodies at the end of the process of oogenesis. Now the question is, why does the mature ovum larger in size, why does the division of meiosis 1 and meiosis 2 in oogenesis is not going to produce equal sized cells? Remember that the oocyte, or that is the ovum, is going to contain all the cytoplasm, reserved food materials and other components that is going to required for the cell to form the zygote after the fertilization. So from the male components, it is not the cytoplasm that is going to come, nor any other components, that is the male nuclei that is going to be received, and not any other components. So to help in that further development, the cell requires the components and some amount of reserved food materials, and all of those are going to be stored in the female gamete only, and that is why the female gamete is considerably larger than the male gamete and also the second polar body. And that is the reason why the divisions of the meiosis 1 and 2 both are unequal division, giving rise to a larger oocyte and ovum compared to the polar bodies. I hope we are clear with these concepts. Now let us compare the spermatogenesis versus oogenesis. There are certain similarities, but there are also considerable differences in these two processes. As you can see that both of these cases, the cell that is going to start is going to be diploid, that is going to be having 46 chromosome or 23 pairs of chromosome, but at the end when you're going to get the gametes, they're going to be all haploid in nature. So in case of the male, the spermatogenesis, the process begins at puberty, whereas in case of the female, the process starts at the fetal stage. And as in case of male, there is going to be mitosis to increase in the number. Similarly, here also you're going to have the mitosis differentiation to increase in the number, and then you get primary spermatocyte in case of male, whereas in case of female you get the primary oocyte. Now in case of male, the process is very simple, straightforward. Primary oocyte undergoes meiosis 1, equal sized cells give rise to the secondary spermatocyte that undergoes the second meiotic division, give rise to 4 equal sized haploid cells, right? That is the spermatozoa. But in case of female, the process is slightly different, as we discussed. After the first meiotic division, it is going to get completed just before the ovulation. So the process of development starts at the fetal stage, get arrested, resumes at the puberty, gets completed. Again, half of that is meiosis 1, gets completed just before the ovulation, giving rise to the secondary oocyte. But the secondary oocyte is going to give rise to the ovum, that mature ovum, and the secondary polar body just after the penetration of the sperm cells.